we are now able to move on to our first panel, and we've got a great one chaired by my dear friend Jay Skyler, an icon in the diabetes world, and one of the best chair people I know. He manages to herd the cats of typical panels like this, where we have extremely smart people, and we're going to do it without presentations. It's going to be a conversation that Jay will get started. So Jay, over to you, and many thanks for your efforts in raising this session.
Well, thank you, Zan, and I'd like to welcome our panelists to join, and ask them to take 1 minute to introduce themselves. I see on video so far Jerry Shulman from Yale. Jerry, do you want to introduce yourself first?
Hi, Jerry Shulman from Yale School of Medicine. I'm an endocrinologist and interested in the pathogenesis of type 2 diabetes and trying to prevent it.
And Hazel, how about introducing yourself?
Got muted. So I was on the faculty at Weill Cornell Medicine, and my whole career has been working on new interventions for chronic diseases. So I've been focused on the mitochondria dysfunction, its role as a root cause for chronic diseases, and have some compounds that are in clinical development. I'm now with a public charity trying to do exactly what Zan was talking about, trying to make available new discoveries to people of all, the entire population. Thank you.
Laura?
Good morning. I'm Laura Niedernhofer, and I'm at the University of Minnesota. I'm a basic scientist in the Department of Biochemistry, Molecular Biology, and Biophysics, and run an institute on the biology of aging and metabolism. My background is really in DNA damage and repair, and thinking more recently deeply about cellular senescence as a target in aging.
Guido?
Good afternoon for me, good morning for you. It's a pleasure to be here. So I'm Guido Kroemer. I'm a specialist in cell stress and cell death, interested in metabolism and aging, and recently interested also in obesity, because obesity can be viewed as an inhibitor of mitophagy, which is the most important anti-aging mechanism. And so the link between obesity and comorbidities may be actually accelerated. And that is what I'm interested in.
And finally, Ralph.
Can you hear me?
Yes, we do.
Oh my God, it's amazing. One of these days I'll learn how to.
We all know that Ralph has computer problems, and has for a long time. And
yeah, he has a brain gap when it gets to turning on the computer. Well, it's good to see all these familiar faces. I'm Ralph DeFronzo, and I'm professor of medicine and chief of the diabetes division here at the University of Texas Health Science Center in San Antonio. And I have a major interest in the pathophysiology and treatment of type 2 diabetes.
Great. So we've heard a little bit about everybody, and they're all interested in the progress toward aging. I'd like to ask the group, and we can go around and let folks comment. And feel free if somebody on the panel raises something that you want to add to just come right in and do that. But my first question is that what do we think are the 2 to 3 targets, metabolic or otherwise, that stand out as likely to leverage multiple chronic age-related benefits? And we've heard a few of you mention the word mitochondria along the way, but maybe take it a little deeper. Let's start with Laura.
Well, one of my favorites is really thinking about senescent cells. So these are very stressed cells. They've become stressed for a number of different internal and external reasons. That results in an irreversible or stable cell cycle arrest, which is a beautiful tumor suppressor mechanism. But the downside to senescent cells is that they have a very potent secretory phenotype that's very pro-inflammatory. And we think that drives the chronic sterile inflammation that is the root cause or of many age-related diseases, or at least exacerbates them. And unfortunately, I think our immune system function as it wanes fails to take these cues from senescent cells and clear them. So top of my list is senescent cells, and I would give a big plug for mitochondria and oxidative stress as well, and just metabolism in general, where we've got a lot of experts here.
Jerry?
Yeah, I'd like to build on Laura's enthusiasm for mitochondria as a target. So I would come at it from the perspective of insulin resistance. So to me, insulin resistance is really what's leading to so many issues with healthy longevity. It's not only metabolic syndrome, it's type 2 diabetes, as we well know. It's fatty liver and NASH, now the leading cause of end-stage liver disease, cirrhosis, cause renal transplant. It's also probably the reason for this huge rise in obesity-associated cancers, insulin resistance, and certainly cardiometabolic disease. So if we can find the root cause of insulin resistance, we can make a huge impact on healthy living. So my view of the mitochondria, and this will get into the basic mechanisms, which I hope we'll get into later, is to promote reverse insulin resistance. And how do you do that? Well, you need to understand what causes insulin resistance. This gets into some of the basic science, but it's ectopic lipid, and specifically certain lipid intermediates. And we'll get into these diacylglycerols. It's a precursor for triglyceride and also things like acetyl-CoA in the liver. But if you rev up the mitochondria, you burn these lipids and you can reverse insulin resistance in diabetes. So I love the mitochondria. I want to rev up, actually make them a little bit less efficient. You'll reduce ROS. I think that'll be healthy for the cell. And more importantly, you chew up, you melt away the ectopic lipid in liver, muscle, these insulin responsive organs, you reverse insulin resistance. And there's a lot of preclinical data. We can talk about that. And I think that will make a huge impact. Thank you.
So Ralph, you mentioned insulin resistance. Do you know anything about that?
A little bit. So Jerry and I go back to the old days at Yale, and it's always great to see Jerry. He and I think, tend to think along the same lines. So I had three things listed that I thought that would be very important. At the top of the list was insulin resistance. So I would agree with Jerry. Lately, with Dr. Roger C. here, we have developed an interest in cardiolipin, and hopefully we'll get to discuss some of what Roger and I have been working on here. And then lastly, what really is sort of driving this epidemic of diabetes is the epidemic of obesity. That clearly is an exogenous, it's an acquired disease. And I think in the United States, we haven't come to grips with the fact that obesity is really a disease. And if you look at people who are overweight, they have an increased incidence of diabetes, as Jerry said. They have an increased incidence of cardiovascular disease. They have an increased incidence of cancer. And I think that if we can develop ways of immobilizing fat out of tissues, either getting it completely out of the body or even redistributing it around. And with regards to the later, I still have a like for pioglitazone. With regards to getting fat out of the body, we have a number of newer agents that will do this. So obesity has really become a scourge. We have over 60% of adults in the US now who are either obese or overweight. And I do believe that what Jerry said is correct, that this lipotoxicity is very, very important in many of the diseases that he mentioned. And I'm sure we'll come up and discuss in terms of diabetes, cardiovascular disease, beta cell failure, etc.
Hazel, why don't you pick up on what your thoughts are for targets?
Yes, well, when I look at the chronic diseases, I see not different diseases with different treatments, but rather what they have in common. So, obviously for all of them, age is a predominant risk factor, but they are all associated with a progressive decline in organ function. over time. And they are all associated with physical frailty, reduced mobility at the end. They have a systemic low-grade inflammation, and it's associated with both metabolic reprogramming as well as tissue remodeling, including the vascular system. So we've been thinking about the major role really of mitochondria function. I think that, what we learned in medical school was just that you need ATP for everything. But I think we've underestimated how much ATP we need. So a little interesting tidbit for the audience is that a human body burns your body weight in ATP every day. The turnover of ATP is 60 to 70 kilograms per day. That's how much it takes. So what is the ATP needed for? It needed for every cellular function, but 75% goes into protein synthesis, protein folding, protein degradation. So the minute you have reduced ATP production, you're going to compromise all of that and end up accumulating misfolded proteins, cell death, and so on. So I've been focused on the mitochondria. Now, to make it interesting, I like the idea of having to control cholesterol with glucose. One of the really intriguing findings that we've had in my work with compounds that promote mitochondria efficiency is that we can prevent or reverse end organ disease without changing blood sugar, blood cholesterol, or blood pressure. So where the mitochondria come in is the role of these biomarkers on the end organ itself. And so that's where we've been focused on. So that these approaches that we have, could be used, together with the preventive measures on the biomarkers.
Great. Guido, you're the last one.
Well, I couldn't agree more that mitochondria are in the center of the world, and that they control the cell's life and death. However, I have a little bit different angle. So the best way to maintain mitochondria healthy is to turn them over. And the only way to turn over mitochondria is to destroy them and to rebuild them. The destruction process is called mitophagy. And it occurs either as a specific phenomenon or in the context of general autophagy. Now, autophagy is a process that is induced by fasting, continuous or intermittent fasting, by exercise, and remarkably enough, by some micronutrients. And autophagy is the process that Mother Nature has invented as the most important organelle turnover pathway that controls cytoplasmic aging. So in model organisms, including in mice, it is perfectly established nowadays that genetic or pharmacological induction of autophagy is delaying aging and improving metabolic health, while its inhibition is causing degenerative phenotypes in virtually any organ and accelerates aging and death. So I like mitochondria, as especially when the uncoupled, inefficient, damaged, and potentially lethal mitochondria are sequestered in an autophagosome and digested to produce ATP, to recycle the macromolecules into micromolecules and energy-rich metabolites and building blocks for new anabolic reactions, and can be hence used also for creating new and healthy mitochondria. So this is my point of view.
Well, it's interesting. Several of you have mentioned mitochondria, but I've heard one person say they want to rev up the mitochondria. I heard another person say they want to promote efficiency of the mitochondria. And now Guido says he wants to destroy and rebuild mitochondria. And are those conflicting concepts, or are they really all saying that what we need is healthy mitochondria that are working, and that's what we need to get to? Who wants to take that? Jerry?
Well, just to clarify, yes, I think we all, I'll speak for myself, but I think everyone is in agreement, healthy mitochondria are important. But again, I want to actually promote a little bit of inefficiency, uncoupling of oxidative phosphorylation. And this, I think, will be beneficial for, again, we've shown both in mice, rats, non-human primates, a little bit of mitochondrial inefficiency will reduce ectopic lipid in liver muscle. And especially if we can liver target an uncoupler, a protonophore, promote mitochondrial inefficiency. You burn the fat in the liver, you defat the liver, you can actually reverse inflammation in the liver, you can reverse NASH. And when you burn fat in the liver, it's better benefit for the cardiovascular system, because you burn fat, you reduce VLDL export, because if you're burning fat in liver, less fat to export, so plasma triglycerides come down, you actually will lower LDL, and you're exporting less fat to skeletal muscle. So you have this knock-on effect to actually reverse muscle insulin resistance and actually insulin resistance in the fat cell. So my feeling is a little bit of mitochondrial inefficiency in liver would make a big difference. We know actually in humans, we reduce liver fat, we can reverse diabetes. We've done this in humans, taken type 2 diabetics, put them on a 1,200 calorie diet for 4 to 6 months, and what their diabetes reverses. And we use isotopes to measure why does the diabetes reverse? Well, it's mostly the liver. Glucose production comes down due to reduced gluconeogenesis. You have enhanced sensitivity to insulin in the liver. It has normal suppression of glucose production. And it's related to this very small pool of lipid in the liver that is gone. So just a 10% body weight loss is enough to melt away all that liver fat. Now that's why nutrition is so important. We talked about that in the previous session. So as an endocrinologist, again, diet and exercise is the best thing that we can do. However, we know it more often than not fails. And so we need other agents to help defat the liver. And just by defatting the liver, we can have a lot of beneficial effects. So just to reiterate, Jay, I want to promote a little bit of mitochondrial inefficiency in liver, and I think that would have very beneficial effects. And will also reduce ROS production. So that will lead to more healthy mitochondria.
And Guido is anxious to get into this discussion.
Well, of course, you're perfectly right. It may be a way to go to render mitochondria slightly less efficient. And actually, the best pharmacological inducer of mitophagy that you use in cell culture is CCCP, which is a notoriously efficient uncoupler that you will probably not give in vivo because it might be toxic. But so one of the mechanisms that causes mitophagy is a reduction of the mitochondrial transmembrane potential. Is actually a whole enzymatic machinery there that waits for mitochondria to get uncoupled or to lose the transmembrane potential, and then will mark these mitochondria that are dysfunctional for subsequent destruction by mitophagy or autophagy. So there's no contradiction between what you are saying and what I'm saying.
So, Ralph, so I don't disagree with what either of the two speakers said, but I'd like to shift sort of the emphasis a little bit to the cardiolipin story, which I think is a very interesting one, which is developing. So for those of people who are listening in who may not be familiar with this story, this is a phospholipid in which two phosphate groups are joined by a glycerol. And then we put 4 linoleic acids on it. And cardiolipin is an incredible, it's a very important part of the inner mitochondrial membrane. And I think it's very clear now in that if you have an abnormality in cardiolipin metabolism, There are very, very, I think, overt disturbances that occur in mitochondrial function. There's a decrease in ATP synthesis. The mitochondria look deranged, and in muscle, particularly skeletal muscle and cardiac muscle, we see skeletal muscle deteriorate. This is frailty, which Laura mentioned earlier. We see heart failure, which has become a big problem now. And Dr. C at our institute has looked at a syndrome called Bart Syndrome. And these are individuals who have a defect in a particular gene that leads to abnormal cardiolipin metabolism. And these people die early in life of heart failure. And interestingly, now there are a number of animal models where it's become very clear that there's a disturbance in cardiolipid metabolism as a function of age. And what Roger has published and what we're starting to look at now is overactivity of alcat1, which leads to an abnormal cardiolipin in a variety of animal models of ischemia induced in the heart. As I said, it's also characteristic of aging, also pressure overload in the heart. So again, this is another type of mitochondrial dysfunction, and I believe that lipotoxicity is going to play also a very important role here in this disturbance in cardiolipid metabolism, which again fits in with what Jerry was talking about earlier. And there are treatments.
Hazel was a real expert on cardiolipid. So Hazel, why don't you pop in too?
Well, Ralph, I'm really glad that you brought that up. I was a little embarrassed to make that a focus, but you're completely correct. I mean, it brings back to the early days of the discovery of mitochondria, where form and the structure and function are completely tied. So cardiolipin, as you pointed out, it has a weird conical structure. It is uniquely different from any other phospholipid. It doesn't make bilayers, it makes them curve. So cardiolipin is what allows the cristae to be formed. And here's another little interesting tidbit. A lot of people are focused on the number of mitochondria, but it's really the cristae density inside the mitochondria that will determine how much ATP you can make, because that's the surface area for where osphos takes place. It's been calculated, actually, that a human body has 14,000 meters square of cristae membranes. And if you can't envision that, which I have trouble, it's the size of 11 Olympic-sized swimming pools. That's the surface area of cristae that we stack into our mitochondria in order to perform properly. Now, with aging, with all the chronic diseases that we are worried about, the non-communicable diseases, there is a loss of cardiolipin content, there is increased cardiolipin peroxidation, and there is a loss of cristae membranes. So that's the energy deficiency that we see with aging. That's what frailty. And I think I agree with Jerry. I think that organ specificity is very important when you talk about this. The liver is very different from the heart and skeletal muscle, where you can't really afford to reduce ATP production by uncoupling. In a very early study, Jerry, I actually work with Ethan Anderson, where he found that with high-fat diet, if you treat with a cardiolipin protecting compound that we had discovered, that you would prevent the development of insulin resistance while the animals are on high-fat diet. And it doesn't change the cholesterol levels, it doesn't change their body weight, they're just as heavy, but they don't develop insulin resistance in the muscle. Okay, so it was very specifically in the muscle. So I think that, I'm sort of like going back to very old literature about the discovery of mitochondria, and the fact that the cardiolipin, another very interesting aspect of it, Ralph, is how the mitochondria has two membranes. The inner membrane is where the cardiolipin is. The inner membrane was the bacteria that we sort of phagocytized. and sort of co-opted it into making our energy supply. But that's all bacteria origin stuff, and it's toxic to us. So that whole mitochondria structure has to be kept intact. The integrity is everything. Once the cardiolipin, mitochondrial DNA comes out, they are perceived as danger molecules. So those are the mitodamps that's getting a lot more attention now. And they would lead to all forms of cell death, as well as low-grade inflammation with inflammasome activation. So, my focus has been on how to maintain cardiolipin, how to increase cardiolipin content in old age and in disease states to restore mitochondria structure. And so far, a lot of work from many people in both animal models and the clinical trials have confirmed that this can be done. We can reverse old age, basically. And definitely, if you start early, you can prevent it. So.
Yeah,
Laura, I'm going to let you. I know that Ralph and Jerry want to react, but Laura hasn't had a chance to react in all this yet, and you're sitting there patiently. Laura, please go ahead.
Well, I'm just struck that it seems to be in this discussion we have a continuum here where we have molecules we need to protect, organelles we need to protect, and then if it gets so bad that you're not turning over your mitochondria producing enough energy, then you get a cell that's very toxic, a senescent cell. And when those accumulate to a certain extent, then you have a diseased organ, because I think Jerry's talking at a sort of a tissue level. And we know that senescent cells are involved in a lot of metabolic syndrome, diabetes. And so maybe thinking about targeting at multiple levels is going to be important.
So where does the inflammation fit in that you all acknowledged was a key importance to this? Jerry?
So I think inflammation is something that comes later. I think it's when in humans, it comes much when you have developed obesity, inflammation, and that will exacerbate things. Studies, preclinical studies we've done clearly have shown that inflammation will then promote increased localized inflammation, cytokines in the adipocytes, more lipolysis, more fatty acid and glycerol flux back to the liver. And if you want to turn on gluconeogenesis, this is how you do it. Fatty acids get oxidized, beta oxidation. The rate of beta oxidation exceeds rates of TCA cycle flux, and so acetyl-CoA builds up. This is a potent activator of pyruvate carboxylase, the key rate controlling step in gluconeogenesis. And then glycerol, remember, for every breakdown of triglyceride, you get 3 fatty acids, 1 glycerol. Glycerol fluxes into the liver. There's no rate control. The more glycerol that the liver sees, the more comes out as glucose. This is the fasting hyperglycemia of type 2 diabetes. And Ralph has well defined this in years past. But this explains the molecular basis for increased gluconeogenesis. And this is why our patients with diabetes present with fasting hyperglycemia and contributes to postprandial hyperglycemia. So, and there's a beautiful study I'd like to just, Roger Davis, it was one of these really beautiful studies, again, in a mouse, where he did a tissue specific, a macrophage specific junk knockout. Junk is involved in the inflammatory pathway. And it was a stunning paper. I remember reading it because everything we and other groups have done, we've dissociated inflammation from insulin resistance in humans. I was very skeptical about the role of inflammation. And even in these young, lean 20-year-olds, this is what I'm particularly interested in. Dr. Peterson, who's my wife, and I, we study young, lean, mostly Yale undergraduates who volunteer for our studies. And we find a distribution of insulin resistance. These are BMI of 23, 24, and some are very insulin resistant, and they're lean. And so we can dissociate insulin resistance, a very important point, from obesity, and it's related to ectopic lipid. It's this triglyceride, more importantly, these SN1,2 DAGs in their muscle, activating pathways that lead to insulin resistance. No inflammation, no evidence of inflammation. And you only see this then when individuals become obese, and then the fat cells become macrophages, move in there and set up lipolysis. But the point I wanted to get back to, Roger Davis he showed these mice would become obese, no fasting hyperglycemia. And that always puzzled me until we collaborated with Roger, sent his mice in. And sure enough, when you study these mice, you fatten them up, there's no increase in lipolysis. So that shows that these macrophages, and later on when you become obese, they move into the fat cell, they promote lipolysis, you prevent that, and you prevent then the exacerbation to fasting hyperglycemia.
Hazel, you had your hand up there too.
Yeah, well, so I wanted to go back to Ralph mentioned, I think why cardiolipin is sort of upstream, the most upstream target for all of this. It's becoming increasingly clear from the literature that cardiolipin is required for mitophagy, because in order to do mitochondria fission, to make them small enough that they could be eaten up, that requires cardiolipin. Cardiolipin is required for mitochondria biogenesis. So you can, if you eliminate all the bad mitochondria, you still need to replace them. And I think that, you know, we need to start looking really upstream and to see all the complex interactions that inner membrane has. It controls. I mean, that was the beauty of the evolution of complex life. You know, how this bacteria allowed us to do things, and in accepting the bacteria, we now have to contend with all the other cellular pathways that get activated when the bacterial origin material starts coming out. And so I think that's where the future study of mitochondria will lie, is how the bacteria and the cell interacts with each other.
Ralph?
Yeah, just one brief comment that I agree with Jerry about the inflammation. I think that's a downstream effect. And then the other thing, there's so many inflammatory pathways that are involved. If you knock out or correct one of the pathways, the likelihood you're going to have a big impact on the aging process, I think, is small. I want to come back again to the cardiolipin story because I've developed a great interest in this. And these diseases that we're talking about, there's an enzyme, ALCAP1, which is overexpressed. And what it does is, rather than putting the linoleic acid so that you have the 4 linoleic acid molecules on the cardiolipin, you have these unusual saturated fatty acid molecules that interfere with the ability of cardiolipin to do its job. And there are a number of inhibitors now of ALCAP1 that have been developed. And there's one that we actually hope to start studies in humans with soon, called dapagliflozin. And at least in the animal models where Roger has looked at it, it is incredibly protective in ischemic cardiac models of cardiomyopathy. It's incredibly effective in preserving cardiac function in aortic constriction models. And interestingly, you know, back to what Jerry has been talking about, it also prevents obesity and also the development of insulin resistance in diabetes. So it has lots of, I think, implications, and there are molecules out there that potentially can restore cardiolipin back to its normal sort of integrity. And these molecules, I think, are look to be safe. So it really, I think we're at a time where we actually can start to look at this. And what I think is even more, I think, encouraging in terms of likelihood to get a positive result, there's a syndrome out there called Bart Syndrome. And people who have this defect in the cardiolipin modeling develop heart failure, as I said, muscle weakness, and they die at a relatively early age. So at least from the cardiac standpoint, we have a clinical model where I think that we might actually have a treatment for this.
Guido, you had your hand up, and then we'll get back, because Hazel's interested in Bart syndrome. We'll get back to Hazel in just a second, but we'll take Guido in here.
So yeah, of course, cardiolipin is a very important molecule. It is, when you generate a lipid missile in vitro, it turns out that cardiolipin is also the one that is most resistant against proton leak. This is one of the reasons why it is an essential ingredient of the inner mitochondrial membrane. Now, it is also a mitodump in the sense that when it becomes accessible due, for instance, the breakage of the outer mitochondrial membrane, or because of an undue translocation from the inner membrane to the outer membrane, it is a receptor for a protein that's called proibitin, and that will guide the autophagic machinery to the sequester and remove that mitochondrion. So that my favorite process is popping up here again. This is one consideration. The other consideration is that we are, of course, confronted with an incredible complexity. So organelles sense stress and emit stress signals. We alluded to mitodamps that then will change the cellular behavior and cause the cell to be stressed, and the stressed cell will emit signals to the environment, and the environment will influence other cells. So it is there's a cascade-like process that can start with the mitochondrion. And I just would like to mention that there's a major, there are several major mitodamps apart from excessive reactive oxygen species and cardiolipin. There's also the fact that mitochondrial DNA can leak out from mitochondria that have undergone membrane polymerization, which is maybe the most severe disruption of the function of this organelle. And mitochondrial DNA then can act directly on pattern recognition receptors within the cell and activate, for instance, interferon responses, activate the inflammasome with the consequent production of interleukin-1 beta that starts off an inflammatory cascade. And inflammation is not just the process mediated by inflammatory cells, specialized leukocyte subpopulations, but it is also a process that can start from a cell that does not belong to the immune system and signal to other cells in the same organ to start what we could call smoldering inflammation or subclinical inflammation that is, of course, an essential contributor to obesity and aging.
Okay. Laura?
Well, all of the descriptors that Guido just used are really characteristics of senescent cells. So it makes you think of sort of this threshold where everything gets triggered and just keeps getting amplified. So we've seen that senescent cells, for instance, are respond to damps. So as you amplify mitochondrial damage and you have these danger molecules in your cytoplasm, this will just amplify inflammation to extraordinary levels. And that then can go on to damage neighboring cells, and that's when everything gets amplified and out of control. So I would still push for some level of thinking about tempering down these senescent cells by helping the body remove some of those so that the damage does not continue to spread.
So we've got cardiolipin in the beginning is one of the instigators. The mitochondria is the central place where it's all happening, and inflammation as a consequence that then creates its own problems and spreads the issues out. Is that a fair summary of what you've all been saying? And we end up with senescent cells?
Yeah. I agree with you, Jay. I think that's a lovely summary of what we've all been discussing. I think that, Laura, perhaps mitochondrial dysfunction is what leads to senescent cells, because clearly without adequate energy, mitochondria not functioning, you're going to have cell cycle arrest. That's clear. And so I think that would be simple. I think that, a quick note to what Guido was saying, the mitochondria damps are now, one of the hot topics that we have to look at. And what's interesting is that with cardiolipin, the oxidized cardiolipin is what translocates from the inner membrane to the outer membrane. The mitochondria doesn't even have to break the outer membrane. It just needs to be sitting out there, and then it becomes a docking station for NLRP3 inflammasome, the whole cascade of inflammasome activation, and IL-1 beta, IL-1 18. So I think that we are only at the beginning of understanding how mitochondria is like the Janus phase, the good and the bad. And we need to learn how to balance and protect the original goal of mitochondria, which is to make ATP. And finally, I think a quick comment to what Ralph mentioned about the Bart Syndrome. I just want to say is that our first cardiolipin protecting compound has been in clinical trials and is now sitting at the FDA for an NDA approval. So hopefully it will become the first example of why protecting cardiolipin could be important for both primary mitochondrial diseases and the secondary chronic mitochondrial diseases.
So we've got a question from Richard Seow of King's College in London, who wants to know, is cardiolipin sensitive to oxidation? And what is the importance of mitochondrial redox homeostasis in all of this? Can we use targeted antioxidants?
So cardiolipin is particularly vulnerable to oxidation because each leg has two double bonds. So yes, it's readily oxidized. What's interesting is that it's not just the superoxide that is produced right there in this environment, but that cytochrome C, we published this, that cytochrome C acts as the catalyst for the oxidation. So cytochrome C is an electron carrier, but it's also a peroxidase, cardiolipin peroxidase. So that's where it happens. Targeted mitochondria, targeted antioxidants with SS31, for instance, has been able to prevent the oxidation. But it's not just reducing the reactive oxygen species, it's really inhibiting cytochrome C as a peroxidase.
So if we're talking now, we sort of moved into interventions. So I want to know, what do we need to harness the targets we've been talking about into interventions that are suitable for clinical trials and moving them forward? And a corollary question is, do we already have products that we are using for one or another indication that may have broader effects and be able to be useful in helping to slow the aging process?
Jay, can I make a comment here? I'm going to turn the discussion a little bit to my favorite drug, which people don't like, okay? And that is pioglitazone, which sort of has a sort of little gray, maybe black cloud hanging over it, but it does a lot of the things that are very positive that we've been talking about. So it's a great insulin sensitizer. In fact, it's the only true insulin sensitizer. But it's a misconception that metformin is an insulin sensitizing agent. Pioglitazone does this. Pioglitazone is very, very good at mobilizing fat out of all organs in the body. And, Jerry said this, in the past, Jerry and I have worked together on this, and we've both gone on. We both believe that lipotoxicity is very, very important. And, there are all these misconceptions. You gain weight. The more weight you gain, what we've shown is the greater the improvement in insulin sensitivity. The more weight you gain is the greater the improvement in beta cell function. So everything you want to measure gets better. The unfortunate thing about pioglitazone is it also stimulates your appetite. And the reason why weight gain is a good barometer is because when you stimulate the PPAR gamma receptors in the hypothalamus, you eat, you gain weight. It's just telling you pioglitazone is doing all these other things. It turns on PGC1, which is one of the master regulators of many of the enzymes that are involved in mitochondrial dysfunction. And then this other myth that it causes heart failure is a major myth, okay? If you look at these people, what you see, yes, they become edematous, but they become edematous because this is a very powerful vasodilator. It generates nitric oxide. And when you underperfuse the kidney, you hang on to salt and water. And the blood pressure drops in the order of 8 to 10 systolic. So there's a lot of misconceptions about what pioglitazone does in terms of fluid retention and weight gain, but it's correcting many of the things we've talked about: insulin resistance, lipotoxicity, mitochondrial dysfunction. So I think there needs to be a lot more focus on insulin sensitizing drugs. And PO, or the TZDs, are the only class of drugs that we have. I don't know, Jerry may want to comment on this.
Yeah, thanks. Total agreement, Ralph. Not surprising. Again, studies we've done preclinically and clinically in humans with at least another TZD, Roussy, and troglitazone showed that, as Ralph says, these are sensitizers. And I agree with Ralph, these are the only insulin sensitizers we currently have. They shift fat. So the reason I would disagree a little bit with it, they're not my favorite, in that I want my patient with diabetes ideally to weight neutral or ideally lose weight. And unfortunately, we're going in the wrong direction, but they do insulin sensitize through the mechanisms that we discussed by reducing ectopic fat. So we've shown liver fat goes down. I know Ralph and Dr. Kusi have shown that we've shown it with Roussy, they showed it with PO. So it's through the mechanisms, lowers fat inside the cell in liver and muscle, and through this mechanism reverses insulin resistance. So that I like about it, but I think there are better ways of doing it down the road, and that's why I like mitochondrial inefficiency.
Who has other potential interventions that they'd like to propose? Guido?
Yes, so we have been working over many years, in particular with Frank Madeo, on the idea that supplementation with polyamines, in particular spermidine, has potential anti-aging effects. It also has anti-obesity and anti-diabetic effects. So there are retrospective dietary studies that suggest that low polyamine intake is linked to an increased mortality from the three major causes of human dying, which is cardiovascular disease, cancer, and so-called other causes that include neurodegeneration. And all of them are positively influenced by spermidine according to these dietary studies, but also more convincingly in mice. So in mice, you can prevent cardiovascular aging, you can reduce cancer incidence by improving immunosurveillance, and you can avoid age-related memory loss and models of Alzheimer's disease to manifest just by giving spermidine in the drinking water. So this is one possibility, which is using a natural compound, which is anyway in our food. The other one is nicotinamide and its derivatives, which are called vitamin B3, that come in different variants and are actually tried to be sold by some companies. I can assure you that the metabolomic studies that we have been doing confirm what is actually known from the literature, that the different forms of vitamin B3, nicotine riboside, and so on, nicotinamide, nicotinic acid, they are interconvertible in minutes in the organism, so it doesn't really matter what you give them. And they have very potent effects also against obesity, against in cancer prevention in some models through an immunostimulatory effect, and against cardiac insufficiency induced by pressure overload, obesity, or aging. So these are two natural compounds that might be used in clinical trials without any fear of toxicity, and that's what I'm selling. Also, it cannot be sold in the sense that there is no patent that will protect the use of these agents.
Others? Laura?
Well, thanks, Jay. Yes, I think there's a lot of room for senolytics. So these are a new class of therapies that really eliminate senescent cells. And I think of these as being drugs that clear cells that are so damaged, you're probably not going to be able to return them to metabolic health. And so they basically play a very pro-inflammatory role and just exacerbate and spread tissue damage. And so there's excellent evidence in preclinical models at this point that senolytics that are clearing these senescent cells work in a variety of different diseases, including type 2 diabetes, neurodegeneration, osteoporosis, cardiovascular disease. And in aged mice, they suppress all-cause mortality, so extending median lifespan, but not necessarily maximum, which I think is very attractive. Another attractive feature of senolytics is that you can give them intermittently, so you don't need drug on board all the time for target engagement. So there's some safety factors there. So I think that that's a good option.
Are there any of them ready for clinical trials?
They actually have advanced to a number of clinical trials already. So these are being tested right now for safety and target engagement. But in idiopathic pulmonary fibrosis, there's already good evidence that senolytics are clearing senescent cells and do have some positive effects. They're in trial for chronic kidney disease, Alzheimer's disease, frailty in elderly women, and now even COVID-19.
Wow. Okay. Hazel, what's your favorite candidate?
Oh, so I really do like the compounds that target cardiolipin. But what I'm also doing now is going to the idea of, natural vitamins and supplements that can also promote mitochondrial function. So we are combining it with selected vitamins and supplements that can promote the TCA cycle, fatty acid oxidation. And I think ultimately it's going to be a mix of things that come directed at different parts of the mitochondria, because the cardiolipin targeting compounds can potentially affect all of the pathways within the mitochondria. But it would be really nice to promote the TCA cycle so that you can talk about enhancing cell repair and cell regeneration for anti-aging.
Jerry, do you have a favorite compound?
Liver-targeted mitochondrial uncoupling. So there are agents that are doing
that's
it.
the target. Do you have a
The target.
compound that
They're in development. I mean, we have, Yale has the patent on it, a controlled release mitochondrial protonophore that does just what we've been talking about in mice, rats, and it's safe in non-human primates. It lowers liver fat, reverses hepatic insulin resistance, that hopefully is marching its way to the clinic. But there are going to be even better, more specific liver target uncouplers that we've developed that we're now testing in preclinical models.
So, if I think about drugs that are on the market that have multiple target organs that they impact, we use them in diabetes and obesity, GLP-1 receptor agonists. Ralph, what are your thoughts on that? Will they also potentially slow the processes of everything that we're talking about?
I actually think that they will. And of course, this is a way of getting people to lose weight. And, I think Jerry and I are big believers that lipotoxicity, fat in your liver causes NASH, fat in your muscle causes insulin resistance, fat in your heart, causes myocardial dysfunction, fat in your arteries causes atherosclerosis. So if you can get people to lose weight, and some of the newer compounds are incredibly potent. And I know for those people, my favorite combination right from the beginning in treating diabetics, and all my patients get stuck on this, is pioglitazone, because it's going to mobilize the fat around. For those people who are concerned about the cosmetic issue of weight gain, you put it with a GLP-1 receptor agonist, and you lose weight, and then you don't have to worry about that. And with an SGLT2 inhibitor, for which there's also some evidence that you actually may promote weight loss and increase fatty acid oxidation. So I think that there is a combination, a triple combination out there that works. And these GLP-1 hybrid molecules are incredibly powerful. We can lose up to 15% body weight So I think that is, sort of like dietary restriction, which many of our patients try, and maybe initially they're successful in losing weight, but on a long-term basis, difficult to people to maintain the weight loss. So the GLP-1 receptor hybrid molecules can be very effective here. And I love them with pioglitazone. I tell people you need to go back and read Proactive, in which pioglitazone was shown to be cardioprotective. And there's only one thing in Proactive that predicts you're not going to die, and that's weight gain. So, when I give a talk, I joke about it, and I said, either you can be fat and dead, or you can be lean and alive. Which you're going to pick? I think I'd rather be a little bit fat and alive. If there was a way that we could sort of look at drugs that would act like the TZDs, but maybe get rid of this cosmetic issue of weight gain, that would be an important way to proceed.
So what do we do in a society where obesity is now being glorified?
Yeah.
We've gone from Victoria's Secret having beautiful young models to having obese people. And you see Old Navy advertising on TV. We've got sizes up to size 30. I didn't even know there was such a thing. How do we, instead of glorifying obesity and saying that, people criticize us if we look at obesity as something that's wrong, they say they're stigmatizing them. They want to make obesity acceptable. How do we get around that and promote that that's really not a healthy way of living, and it causes so many diseases and is responsible for all these things? How do we change the messaging?
Well, I think in our society, we haven't come to the point that we accept obesity as a disease and understand it as a disease. I mean, the more overweight you are, the more likely to have a heart attack and die. The more overweight you are, the more likely to have a stroke. The more overweight you are, the more likely you are to develop diabetes. Every organ is adversely affected by obesity. And I don't think we should stigmatize people. I think that's the wrong way to go. I just think that we need to educate people that weight gain, and I think all of us who are here in this panel understand that, okay, it's not so much the weight gain, it's the fat within the tissue that's doing all these bad things, that we need to promote weight loss. And the other disturbing thing is we're seeing this enormous epidemic of obesity in our children, and the data are very clear that childhood obesity, this is what's leading to obesity in adults. And it's really a big problem. And I think, we have drugs that actually can help, but it's difficult to get people just from dietary interventions alone, outside of sort of these clinical research settings, which I think really are not very practical to get people to lose weight and keep it off. So there's got to be a big educational process here. Other people, Jerry.
I just don't think we should be glorifying obesity like what's happening on TV these days. But Jerry?
I just want to make a point that it goes to, in my view, beyond obesity. Lean individuals have insulin resistance. Again, bringing us back to the initial discussion of the pathogenesis of cardiometabolic disease and obesity-associated cancers. Insulin resistance is what we need to fix. We've dissociated obesity from insulin resistance in young, lean 20-year-olds. We see insulin resistance, profound insulin resistance and ectopic fat in our participants from India. Asian Indians have, again, BMI 22, 23, profound insulin resistance, and they're thin as can be. So we have to be thinking, I think, beyond obesity. And again, it's related to, again, this mechanism of ectopic lipid. And again, I think the best example also of this is the lipodystrophic individuals, either acquired or inherited. Again, thin as can be, and profound insulin resistance, diabetes, very difficult to control, and it's related to ectopic fat in liver and skeletal muscle.
So I just want to, I agree with Jerry, but I want to take it a step further. So actually my first NIH grant, if I can think back that far in 1975, was just to look in the normal population. And what we showed was that 25%, as Jerry's saying, of the normal, quote, lean population is severely insulin resistant. So I'm a big believer that insulin resistance, and in fact, there are 17 prospective studies that show if you make a measurement of insulin resistance and you follow these people up, insulin resistance in 17 out of 17 studies predicts who's going to have a cardiovascular event. So clearly insulin resistant in and of itself is a big problem. But then as you pointed out, Jay, I think that obesity is just a separate issue, is that this is an acquired defect, okay? So you can take people who are normally insulin sensitive and they now start to gain weight and they put fat everywhere. So this is an acquired form of insulin resistance and lipotoxicity. So I think they're both very, very important.
Jay, I just wanted to jump in. First of all, this discussion is just beautiful. It brings tears to my eyes as a student of metabolism. So I wish it could be a day-long symposium. And we do still have 15 minutes left, so I'm not by any means suggesting we need to hurry. Of course, we are going to have a panel on behavior and addressing obesity from that standpoint. It's going to be very robust, so we look forward to that. I do want to come back to Ralph. Ralph, you've touched just about every major diabetes drug class in its development, and metformin is a good example of one you certainly had your fingerprints on. And that has been put forward as another agent that has a relatively safe profile. And so that is often thought of as a candidate. But going back to pioglitazone, let's keep in mind that we're talking about starting people relatively early on, let's say 30 years old. And for people with a potential for osteoporosis, pioglitazone could be an issue in that respect. It doesn't have a clean set of sort of mechanistic impacts on the body. And so that would be a concern, just generically, that there may be a number of products that look promising, but they do need to have a clean safety profile.
Yeah, well, as you pointed out, I think the major issue is with pioglitazone is the bone issue, not the other things that people are, I think, mistakenly concerned about. Remember, pioglitazone, the primary problem in terms of the bone issue is in postmenopausal females, and it's dose related. So the signal, if it's there, is weak in men and premenopausal women, and if you stay less than 30 milligrams. I personally been using this drug for forever and ever. I'd never seen a bone fracture. Now, I don't deny that out there in the literature, but I tend to stay at 30 or less. You know, we did the dose response with pioglitazone, I don't know, 2001. It's published in Diabetes Care. The drug probably should start at 7.5 milligrams, go to 15 and 30 and not above. But if we're concerned about, like, as you point out, the bone issue, then we still have a good insight. The drug is doing something very good. So are there ways in which we can focus on, let's see if we can develop molecules that will take the good aspects of pioglitazone and divorce it. The major problem, in my opinion, is going to be the bone issue. Now, with regards to metformin,
don't forget that we had signals in other members of the class in animal models of carcinogenicity. So that's another theoretical concern, that over many decades, there could be an increased risk of cancer. Not necessarily with pioglitazone. And I know it's a complicated issue, but
I mean, yeah, we could spend all day looking at the story of pioglitazone. I personally don't believe that pioglitazone causes bladder cancer. And I think the FDA mandated study in Kaiser Permanente pretty much sort of should clear this up. In these meta-analyses, 70% of, first, meta-analyses going back on these databases really are a poor way to look at pioglitazone in bladder cancer. And even if you want to use them, 70% of the meta-analyses are negative, saying pioglitazone doesn't cause bladder cancer. But again, there are ways. We know that pioglitazone, it activates PGC1. This is the master switch for mitochondrial biogenesis. Can we mimic this with another molecule that may not have these concerns? So we should take the message and say, look, pioglitazone does all these good things. Yeah, maybe there's some bad things that it does. Can we capitalize on the good things? Now, in terms of metformin, and as you pointed out, I brought metformin to the US market in 1995. Metformin is a very good drug. And of course, in 1995, when we only had sulfonylureas, this was a godsend. Now we had metformin SUs and insulin. But we have many, many better drugs than metformin today. Now, Jerry is one of the leading experts in terms of mechanism of action of metformin. It works primarily on the liver. But, you know, we showed when we did this development for the FDA that metformin was not an insulin sensitizer. And metformin gets into cells through the organic cation transporter. And that transporter doesn't exist in muscle, either skeletal muscle or cardiac muscle. So it can't possibly get into muscle. So to me, it was a no-brainer from the beginning. It couldn't be an insulin sensitizer. Now, is it a good drug for treating diabetes? Yes. Does it work through the liver? Absolutely. But is it likely to do a lot of the other things that people are talking about in terms of anti-aging, et cetera? I have to say I'm a little bit skeptical.
There's a whole panel that's going to discuss that later during this this Metabesity conference. So I think we ought to abandon that and look at other things here. And one question for Hazel was, what do you think about resveratrol?
I don't have any insight into this question. So I won't take time for that.
Anybody else have any insights into that as to whether it's...
Personally, I think this is another molecule that's overhyped in terms of its benefit. But that's my personal opinion. Maybe one of the other people want to comment on it.
Guido?
The question was on resveratrol, right?
Yes.
So actually, when you give resveratrol to mice, this is published by other groups. They tried desperately to show that it increases longevity. It does not. The only thing that resveratrol, in contrast to, for instance, spermidine, obtains is a reduction in morbidity under the circumstances of high-fat diets, but in normal diet, it has no positive effect on the health of mice. So it's probably not the very best candidate to. There are better ones.
Now, Guido, you live in Paris, and red wine has been raised at.
Yeah, I'm all for it.
Actually, we published a small study years ago on polyphenols that you can find in red wine and that you can buy from Sigma or other providers. And so we investigated their autophagy-inducing capacities. There were actually several polyphenols. And more interestingly, even there are combinations of polyphenols in red wine that are having hyper-additive synergistic effects. So it is not just about resveratrol. In red wine, there are much better things than just resveratrol.
Bordeaux or Burgundy better?
I
prefer Burgundies, but that's a personal preference.
The Côte d'Iron in my case. No, the content of polyphenols in different areas of wine and different strains of wine in different on the same location in the shadow versus the sun are extremely heterogeneous, and this is something that has not been really well investigated. So it might be interesting to do a sort of systematic mass spectrometric quality control of red wine to judge it apt or not for improving our health.
I suggest that we all meet in Paris and take a boat trip and then through the wine country, and then we can actually do a scientific tasting
Exactly.
and then we'll follow us all up to see which one of us lives longer.
Exactly. And we don't have the problem of glorification of obesity yet. Because thin people are still the majority.
And so what about chocolate and cocoa and its potential benefits? Anyone? Okay.
I can only say I
No,
believe that.
Yeah, I'll take that, Jay.
I like that.
Yeah, we all love chocolate. And actually, I'm high on flavonols. I think that there is promise for what they do, certainly for cardiovascular function. And as you may know, we're going to have a readout coming up pretty soon of the Cosmos trial, which is looking at high-dose flavonol effects on cardiovascular and other, let's say, members of the Metabesity constellation. So that may be something that is helpful, at least for cardiovascular health. There's also a lot of evidence that it helps with cognitive function. And so keep your eyes on that. We know that it does have good effects on blood pressure. That's likely to at least translate into a CV benefit.
So we should serve chocolate during happy hour with our wine.
Well, in moderation.
Or even
As long
better,
as we don't get obese, right?
That's
even
right.
better, I might recommend chocolate covered strawberries because the flavonoids in strawberries and other fruits are senolytic and quite potent. It's just we can't get quite enough from food, unfortunately.
Interesting. Are there other additives, nutraceuticals, nutrients, things that we can use to help aging that anybody would like to suggest?
I wanted to hear about Jerry's compound at Yale. Is that something that we should be looking for in a pharmacy in the next 10 years?
So I think it's going to work, and I think most importantly, it's going to be safe. I mean, we have safety first, especially for any agents going to be used chronically. But and that's why we're marching through all these preclinical studies to establish the safety as well as efficacy. But again, it comes down to the root cause of insulin resistance, which leads to all the other things. And we didn't get into the actual basic mechanisms. I'm happy to do this, but it relates to lipid metabolites that actually lead to a block in insulin signaling. You have a block in insulin signaling, and you have insulin resistance. I just want to make this an interesting point about insulin resistance. Why does it exist? Why through evolution? And so the work that we've done, I think, gives some answer to this. So when lipid builds up, these SN12 DABs activate PKC epsilon that phosphorylates the threonine on the insulin receptor itself, right in the kinase domain. This threonine sits right next to two tyrosines that are required for autophosphorylation, and it's conserved from humans down to fruit flies. So during starvation, all animals get ectopic lipid, including humans. We all get fatty liver during starvation, mice, rats, humans, with prolonged starvation. Insulin resistance is beneficial to protect us against starvation. And now in our Western environment, this is leading to cardiometabolic disease. So this is, I believe, at least my view of why insulin resistance has persisted. And this threonine specifically has persisted throughout evolution.
Wow.
Wonderful. Wow.
So what we really want to do is get everybody to be thin, to be physically active, potentially to have intermittent fasting, to drink wine with their chocolate covered strawberries in the evenings, and try to get plenty of sleep and do well. And with that, I think we've had a great discussion. I'd like to thank our panelists. I'm going to turn it back to Zan and Larry. And thank you for having invited us all to be here today. I think I speak for all the panelists that we've had, I think, what has been a lively discussion.
Thank you,
Jay, and
everyone. Thank you.
Now, bye-bye.
Bye.
I just can't thank you enough, Jay and dear colleagues. That was just amazing. Again, for me, I had tears in my eyes because of the insights that were coming forth. And so we will continue with you tomorrow, Jay. You're going to pick up another panel that will look a little further down the development pathway, and that will be equally valuable.